Hi, hello everyone guys. So today I am making the part 2 of the video Cell the Unit of Life. Uh, in this video we will learn only the prokaryotic cells about not in very detailed. So moving to because prokaryotic cells do not contain membrane bound organelles, we know that one. So it will comes on the eukaryotic, which is in an animal cell, and a plant. So let start the prokaryotic cell, which is represented as a bacteria and blue-green algae, also called as a cyanobacteria, mycoplasmid, which is in a smallest organism, smallest bacteria we have mentioned in the previous video, which has a very small size, mycoplasma, and PPLO in a short form. In a abbreviated form we write this one. The full form it is pleuropneumonia like organisms. So here we know they may vary greatly in size and shapes, but in the coming to bacteria, it varies in size also and shape also. The four basic shapes of bacteria are bacillus, which is a rod like, and coccus spherical like, vibrio comma shape, and spirillum is a spiral. Next coming point is that all prokaryotes have a cell wall. Yeah, not means like it didn't have a cell wall. It have a cell wall, but except it's in a mycoplasma. It do not contain double membrane like a cell wall, plasma membrane, as like that one. But all prokaryotes have the cell wall except the mycoplasma. It is in a without leaving cell wall, and having a fluid matrix called a cytoplasm. And in prokaryotics, they do not have a well defined nucleus, and the bacteria have the small circular DNA outside genomic DNA. So genome, we usually know the group of the genes are called genome. Genomic means we have learned the which we learn the structures and parts and the complexities of the genes, that a group of genes is called genomic. So here the bacteria in that small circular DNA outside the genomic DNA is present, and these smaller DNA is called as a plasmid. And that plasmid also like it is resistant to antibiotics, and it confers certain unique characters of phenotypic to such a bacteria. And also it's having a plasmid DNA. It is used to the monitored bacterial transformation with foreign DNA. There are some important points of about plasmid which may be asked in a neat paper by MCQs. Next, a specialized differentiated form of a cell membrane means it's like a infolding of a cell membrane called as a mesosomes, and that infoldings or extensions, it is in the forms of vesicles, tubules, and lamellae. Next coming is cell envelope and its modifications. How the prokaryotic cells have its cell envelope and its modifications. Coming to bacterial cells have a chemically complex cell envelope that consists of tightly bound three-layer structures. That outermost layer is called as a glycocalyx, which is followed by cell wall and then plasma membrane. And this glycocalyx includes glycoproteins and glycolipids, and thickness, it could be loose sheet, that is called as a slime layer. In other thick and tough, it is called as a capsule. Next coming to cell wall, it determines has a shape of cell, and it provides strong structural support to prevent bacterium from bursting from it means also like a protecting. Next is plasma membrane. It is a selectively permeable in nature and interact with outside the world. It is the outermost layer, cancel that one, out of the cell wall. It is selectively permeable in nature and interact with the outside from coming not to inside for protecting the cell. Mesosomes is an extension of vesicles, tubules, and lamellae. It's a forms of we have told in a back that one. So that one which is vesicle, tubules, and lamella helps in the cell wall formation, DNA replication means copies of DNA, and distribution to daughter cells. Helps in respirations, secretion processes, and also mesosomes that forms helps increase the surface area of the plasma membrane. It will has the in the inside the cell the newly mitochondria, or means mitochondria will be there in prokaryotes. Some of them is rare, so which get synthesized in the cell. Then it have a space, it have to be in a space. So the plasma membrane also it increases the surface area and enzymatic content. In cyanobacteria, membranous extensions into cytoplasms called as the chromatophores, which contain pigments. And if bacteria motiles, they have thin filamentous extensions from cell wall called as a flagella, uh, and it composed of three parts: filament, hook, basal body. And this flagella role is in motility. So beside flagella, pili, fimbriae, and also surface structure of bacteria, but they do not play role in motility. Next, coming to the now I tell that it's a flagella content, just motility. And like in bacteria, there is a types of flagellations like a trichus. From A we can say absent means no flagella. Example is lactobacillus. Monotrichus, mono means one, which means one flagellum. Its example is Vibrio cholera. And Lopotrichus, which means a flagella, it having the group of flagellus or one flagellum at the one end called as a spirillum. And Amphitrichus, which means it's having a group of flagellas at the two ends also. Example is Nitromonas. And Peritrichus, peri means many, so many flagella are present. Many flagella full of the cell. Example is E. coli. Next, pili. So it's also like an activity is like an flagella, but it is an elongated tubular structures made of special proteins, and the fimbriae are the small bristle-like fibers sprouting out of cells. Coming to ribosomes, it's a non-membrane bound organelles. So in prokaryotics, the ribosomes are the 70S. Here S means it's one of Svedberg unit. We said it's a coefficient which is made up of two subunits of 50S and 30S. Svedberg unit, S means, and they are about 15 nanometer by 20 nanometer in size. And ribosomes are the sites for the protein synthesis, and many ribosomes are attached to mRNA. It is a process of the translation. So here many ribosomes attaches to the mRNA called as a polyribosome or polysome. Two also equal, just synonymous. And the ribosome of polysome translate the mRNA to protein. Either this we seen in the central dogma, which is next in the second year. And reserve material in prokaryotic cells are stored in cytoplasm in form of inclusion bodies. So inclusions means it is an unavailable, like an it means uses for not a living like other types of, which is lies in free inner cytoplasm, and they are not bound by any membrane. Like example, phosphate granules, glycogen granules, synovial granules. And also like a gas vacuole, also it's found in blue, green, purple and green photosynthetic bacteria. These are food materials in also, but gas vacuoles only we seen in the blue, green, purple and photosynthetic bacteria. So these end this chapters here. Prokaryotic cells we learned. In the next part of the video we learn the eukaryotic cell and the membrane bound organelles detailed structure studies, which is very interesting. I giving the tricks for that video. And thanks for watching this video, and do not miss to like, share and subscribe the channel. Thanks for the watching.